Hello, everyone. I'm Daniel Martinez-Krams, and I am pursuing biology with honors at Stanford University. During my time here, I have conducted research in the Majetti Lab studying acute myeloid leukemia, or AML, an aggressive blood cancer with poor long-term patient outcomes. Today, I want to present to you my project, Mesenchymal stromal cells rescue hypoxia-induced survival defect in ex vivo acute myeloid leukemia cell cultures. Necessary background for this work is the process of hematopoiesis, or the production of blood cells, and an overview of ongoing research efforts, cell culture model systems, and the push to recreate the in vivo microenvironment. During healthy hematopoiesis, hematopoietic stem cells can both self-renew to create more hematopoietic stem cells or start a differentiation pathway that leads to every blood cell. These hematopoietic stem cells reside and self-renew in the bone marrow niche, supported by endothelial cells, osteoblasts, and mesenchymal stromal cells, or MSCs. When hematopoietic stem cells differentiate, one of the first divisions is between lymphoid cells, your B cells, T cells, and natural killer cells, and myeloid cells, such as monocytes, erythrocytes, and megakaryocytes. AML is a disease of the accumulation of early myeloid cells, as hematopoietic stem cells acquire mutations and become malignant leukemia stem cells. Thus, AML research has often compared healthy hematopoiesis to its malignant counterpart. This has led to a focus on the niche, which contributes to AML relapse and drug resistance. It is hypothesized that this pro-leukemic effect is facilitated both by niche cells and the niche context. For example, bone marrow oxygen partial pressure is approximately 5%, far lower than the 20% oxygen in ambient air. AML studies, however, are limited by the available tools, in vivo and ex vivo models. In vivo mice models are able to replicate both cell-dependent and cell-independent niche effects, but are expensive and time-consuming. The push toward personalized medicine will require a fast but accurate model of the patient's AML, such as an ex vivo culture system. This may become increasingly popular as new AML drugs are approved and clinicians seek the optimal drug or combination of drugs for an individual patient. Currently, ex vivo culture models have incorporated either co-culture with niche cells or hypoxia, but rarely both. Thus, I sought to develop an ex vivo co-culture system with AML patient sample cells, physiologic hypoxic conditions, and MSCs. Then I compared the results of the ex vivo culture system with primary AML and MSC expression data. First, I showed that hypoxia decreases the viable cell number of AML patient sample cells. I cultured patient sample cells in media containing serum or a serum-free alternative with polyvinyl alcohol and ITSX. In both cases and for both samples, growth decreased in hypoxia. Then I showed that MSC co-culture can rescue this decrease in cell number and viability, and that this occurs in a non-contact dependent manner. Patient sample cells were added directly on top of the immortalized mesenchymal stromal cell lines UE7T13 or HTERT, or added to a transwell insert above the feeder layer. I also included wells with media only and wells with media and human cytokines as control conditions. In hypoxia, AML MSC co-culture with or without direct contact increased AML cell viability compared to cytokine supplemented media. This led me to show that the AML cell-supporting factor is stable and contained within the media. I stained AML cells with cell trace dye, which dilutes after cell divisions. After 3 days, conditioned media from UE7T13 or HTERT MSCs supported AML cells as well as media with cytokines. Finally, I shifted to comparing in vitro results with in silico analysis. I collected MSC conditioned media after 5 days and profiled the cytokines with a Luminex assay. I also analyzed the expression of cytokine gene transcripts in primary sample MSCs from AML patients and healthy donors. I found that MSC conditioned media contained cytokines that are also upregulated at the transcriptional level in MSCs. To close out this computational analysis, I looked at RNA expression in AML cells. I used RNA sequencing data from sorted leukemic populations of 12 AML patients and sorted hematopoietic stem cells from 4 healthy donors. I found that healthy and leukemic hematopoietic cell subsets cluster by the expression of cytokines and cytokine receptors. Overall, I conclude that bone marrow physiologic hypoxia decreases AML cell growth, which can be rescued by AML MSC co-culture or MSC conditioned media, but not cytokine supplemented media. I also found that MSC conditioned media contains cytokines that are transcriptionally upregulated by healthy primary MSCs, and that cytokine and cytokine receptor expression clusters cells in the healthy and malignant blood compartments. Importantly, I developed a clean and robust model for ex vivo AML studies. This is valuable because of AML patient sample heterogeneity, which is both a limitation of the study, but also a benefit to using an approach like mine. This study motivates future research to determine the MSC-produced soluble factor or factors responsible for rescuing the survival defect in hypoxia. Additionally, it provides a platform for niche-like ex vivo drug screening and personalized medicine. Thank you so much for listening to my presentation.